So with that, I'm really happy to introduce our first speaker, David Baker, who is joining us from the University of Washington.
All right, I'd like to thank the organizers for inviting me to be here. Let's see, how do I get the first slide? Do I just? Oh, black key? All right, here we go. So I'm going to tell you today about designing proteins from scratch. Natural proteins really span a very tiny fraction of the sequence space that's available to proteins. So there are 20 amino acids for 100. So for a 100 amino acid protein, there's 20 to the 100th possible sequences. And I've depicted that with this big gray box here. And in this space, nature sampled these little red families, which occupy really a tiny, tiny fraction of this space. So maybe 10 to the 15th different proteins have been sampled during evolution. There's 10 to the 130th possibilities. So really, nature's just barely scratched the surface. So almost all protein engineering to date is what we call Neanderthal protein design, where you take something that's lying around in your environment, like a naturally occurring protein, and modify it a bit. So I'm going to tell you about today is de novo protein design, where you build proteins from scratch from first principles. So the basic concept underlying this is that proteins fold to their lowest energy state. So if you're trying to predict the structure of a naturally occurring protein, you take its sequence and you search for the lowest energy state on a computer. If you're trying to design a brand new protein, then you have to come up with a sequence whose lowest energy state is the structure that you're interested in making. Now, I'm not going to tell you in detail about the computational model, how we calculate energies, but it's kind of what you'd expect. There's van der Waals interactions, hydrogen bonding, electrostatics interactions, and so forth. And the model isn't perfect. And so what I'm going to first start off by telling you is how do you improve computational models in this area? And I think there's sort of a general application of computational models in biology generally. And what you really need is a lot of data. So you need to design lots and lots of proteins, completely new proteins, and then see which ones work and which ones don't, and then update your computational model accordingly. So Gabe Rocklin, a postdoc in the group, developed a method for both computationally designing and then experimentally testing for folding not just one small protein shown here, but tens of thousands of small proteins at a time. And so he could carry out large-scale design calculations, pick out on the order of 10,000 different sequences predicted to fold into very low energy states, and then test them all experimentally. And he started off with these very small proteins, and these are a real challenge for protein design. They're 40 amino acids. It's really hard to get a stable protein with just 40 amino acids. And so in the first round, when he tested his 10,000 designs, he had relatively few successes. But he was able to learn from the successes and failures, basically do machine learning on the data, improve the computational model, and then through basically repeating this, sort of improve the model, do a large-scale design calculation, collect experimental data, and then iterate, he was able to get to the point where a large fraction of his computational designs actually folded into stable proteins. I'm not going to show you all the data these proteins fold, and that they fold to their correct structures. You have to trust me on that. But the upshot of it is not only do we have a much improved computational model, but now I told you that there were very few naturally occurring proteins in this size range. In fact, Gabe synthesized them all. This is using high-throughput DNA synthesis Agilent oligo chips, so we can encode a 40 residue protein on a single long oligo. So we could actually test all of the naturally occurring proteins, and this is increased stability here. So you can see, these colored bars represent Gabe's de novo design proteins. So the most stable things he tested are up here. You can see there's only very, very small number of naturally occurring proteins which are very stable. And now we have this vastly larger number of completely designed de novo proteins. And this is really where we're going. We believe that in 10 or 15 years, basically all the proteins people use for in technology and other areas will be designed proteins that were designed to have the properties that one wanted rather than just adapted from something which happened accidentally during evolution. So we can go smaller. If we go smaller, we have to put we have to make the problem more constrained. So these are circular proteins. On the computer, it's no problem making circular proteins. Here we have to chemically synthesize them because it's very hard to get organisms to make circular proteins. And these are comparisons of these designs to NMR structures. You can see we can design them very, very accurately. They also turn out to be extremely stable. It's basically impossible to denature them. And we can go even smaller to make things that are like cyclosporin, macrocyclic compounds, which have the nice property that we can essentially enumerate all the stable states that you can have in a macrocycle. Again, these are comparisons of NMR structures to design models. So you can see we can compute these structures on a computer and then make them in the lab, and they come out almost exactly the way we designed. These are exciting compounds because they can be potentially orally available, like cyclosporin. So we're currently working on how to get them to get across membranes and target targets of interest. Okay, so we can take this methodology for designing new proteins and design proteins to bind to targets of interest. So here's the influenza hemagglutinin here, and here's the botulinum toxin here. And what I'm showing is a little designed protein that's been designed from scratch to have shape and chemical complementarity to the virus. And this is actually a crystal structure showing a superposition of the designed protein on a crystal structure of the designed protein in complex with the viral protein. And the same here. This is a crystal structure of the botulinum toxin binding protein bound to botulinum toxin. You can see that in both cases, the computational model is almost exactly right. So these proteins neutralize the flu and the toxin, and they have some neat properties compared to antibodies. You can heat them up at 80 degrees, and they don't lose any binding activity, whereas if you take a neutralizing antibody, it quickly loses activity. I'm not going to go through the data, but these small proteins provide both prophylactic and therapeutic protection in animals against the flu. So we're really excited about this as a new class of therapeutic. These proteins can also be, of course, designed against host proteins. So this is a small protein we've designed against alpha V beta 6 integrin, which is commonly upregulated in tumors. Here's a mouse that has a single tumor that is alpha V beta 6 positive that's been injected with a fluorescent version of this molecule. And you can see that after about 20 hours, the only place where you see this molecule in the mouse is in the tumor. So we're quite excited about using these reagents for targeting. For stem cells, we're working with Hannu Ruohola Baker's lab to design proteins that bind to intracellular protein targets that allow disruption of complexes of interest. So that could be interesting for the organelles that Rick was just mentioning. So here's a design protein that binds to EED, a component of the polycomb complex. And when you express this protein under the control of an inducible promoter, you quickly abolish histone methylation. So this is when you express the protein, and and it causes stem cell differentiation. So you can see here what this is the binding protein. When you express it, it disrupts the polycomb complex. We can also design proteins that bind to small molecules. So here's a de novo designed protein, again, compared to a crystal structure. You can see it's very accurate. And we've designed it to have shape and chemical complementarity to the GFP chromophore. So now, in this case, the chromophore is being added to the cells afterwards. It's not constitutively expressed, which makes the fluorescence have some very nice properties. So we get this huge fluorescence induction when the protein is when the protein is added to the small molecule. So we're pretty excited about the possible uses of this for cellular localization. I'd be happy to talk to people here about that. We can also design proteins that bind to protein small molecule complexes in such a way that the complex formation is absolutely dependent on the small molecule. This is work with Glenna Foyt and Dustin Malley. And in fact, in this case, we have different proteins that bind to recognize different small molecule protein complexes. And we can use this to make, in this case, a transcriptional activator whose activity is dependent on which small molecule is added to the cells. So this is very interesting for synthetic biology. It's basically a whole class of new orthogonal heterodimerizers. Now, most proteins are held together by hydrophobic interactions. Since we're designing proteins from scratch, we can design proteins based on different principles. So in DNA is a very nice property that's held together by Watson-Crick base pairing. So it's very easy to design a complementary piece of DNA to one that you have to start with. And nature doesn't do this with proteins, but we wanted to be able to do it. And we've made quite a bit of progress. So what we're doing now is designing proteins whose cores are not hydrophobic, but they're held together by very detailed hydrogen bond networks between amino acid side chains. There's actually a superposition of a crystal structure and a design model. And you can see that these interactions are coming out almost identically to those designed. So this lets us do some very nice things. We can now generate whole families of interacting pairs just in the same way you do with DNA. You just change the order of the bases. Here we can do the same thing, but with proteins. By putting different hydrogen bond networks at different layers, we can get different specificities. And we can extend this beyond dimers to, in this case, is heterotrimers. So here are 3 proteins with a hydrogen bond network that is only satisfied when you have all 3 chains. When we make these proteins in a cell and put a tag on one of them, you pull down all 3 bands. And if you look at what's formed by native mass spectrometry, you only find the complex. So this is really good for 3-way AND logic. You need all 3 proteins to get the complex to form. We can also use this concept to make completely new membrane proteins. So here's a de novo designed membrane protein, which is held together by hydrogen bond networks in between these helices. Here's the design model, and here's the crystal structure of the protein that we've been able to extract from cells, and you can see a superposition here. So this is really interesting for making new classes of receptors, for example. We can also use this concept to design switches. So here again, 3 chains that are held together by a hydrogen bond network, but in this case involves histidine residues. And histidine residues, when they get protonated around pH 6.5. And you can see what happens if you protonate this network. It should completely blow things apart according to the model. So if we make this protein, it forms a trimer above pH 6.5. But when the pH starts to drop, it comes apart and forms a monomer at low pH with some intermediate species at intermediate pH concentrations. So this is basically a pH-dependent switch. What's cool about it is that when it opens up, there's still some hydrophobic surface here. And so if you add it to liposomes, at pH 7, nothing happens. But as this thing opens up, it starts punching holes in liposomes. And this is a dye quenching assay. All the dye rushes out. So we're very excited about this for endosomal escape. So when this protein gets taken up into endosomes, it should come apart and hopefully permeabilize endosomes. We can also use this concept to design protein allostery. So here we've designed a protein that has a binding peptide. This is for BCL2, the apoptotic protein, anti-apoptotic protein. And this peptide is completely caged. So if we put BCL2 down on a chip, this doesn't bind at all. But if we add a key, which has better hydrogen bond networks and can basically displace this, if we add it, then interesting things happen. If we add a short key, then it's too short and this thing stays closed. There's no binding. But if we add this very long key, then this epitope gets pushed out and it can now bind BCL2. And that's shown here. So this gives absolute 2-way AND logic. You only get binding to BCL2 if you have this protein and this protein. And we're very interested now using this to get selective AND-like logic for recognizing, say, tumors that have 2 targets, 2 proteins on their surface that are found also in other parts of the body but never together uniquely. So we can use this scheme in vivo in cells. This is with Hannah El-Samad's lab at UCSF. In this case, what's being caged is not a BCL2 binding motif, but a degron sequence. When the key is not expressed, this is a perfectly happy transcription factor. You get more, as you increase its levels, you get more and more transcription. But when you express the key, you displace the degron, and the whole thing gets degraded, and you get less transcription. Okay, so the last few minutes I'm going to talk about design of assemblies. So these are 2 proteins that have been designed to self-assemble into these icosahedral nanoparticles shown here. And we can make quite a wide range of different types of nanoparticles and EM images. We have a his tag on one of them. We can purify them, and you get quite different structures. We've been able to solve structures of 3 of them, and they basically come out almost identically to the design models, which is pretty cool. I showed you that we can design 8 residue peptides and get them accurate. These things now are huge. They have thousands of amino acids, 163,000 heavy atoms in this case, and they come out exactly right. You can see the EM averages compared to the design models shown here. Okay, so the last thing I'm going to tell you about is we can take these capsids and we can modify their interior surface so they package their own genomes. And so when these are made in E. coli, the proteins in the mRNA assemble, and so you get a particle with its own messenger RNA inside. So if we generate a library, then we get a whole collection of different particles with different genotypes inside, and then we can carry out selection of different sorts. This is evolution, but starting with a completely synthetic molecule. The initial particles barely protect their own genome, but after selection in ribonuclease or blood, we get things that can protect their genomes for quite an extended period of time. And if we take these particles and make a library and put them into a mouse, even these ones that are stable in blood alone get quickly removed from circulation. These particles that are, if we put them into mice, pull out blood after 4 hours, and then repeat a couple times, we can find particles that circulate for very long periods of time in mice. So we're very excited about these for targeted delivery. And so it's basically what I wanted to tell you about today. We show you that we understand the fundamentals of protein and assembly, and we're really poised now to build a whole new world of synthetic proteins. And we're really interested to applications to cell science and other areas. So if you have any ideas, please let me know. And thanks.
So David, great talk as always. One question I have, when you make purely synthetic proteins that you introduce into the body or into whole animals, do you evade the immune system or do you potentiate the immune system? Or could you do either on a case-by-case basis?
Yeah, well, let's see. So we've looked at that most extensively with that flu-neutralizing protein. And the experiment that we did 2 experiments. We first immunized animals repeatedly with the protein and looked for an immune response, and we didn't detect one. But more critically, we exposed mice over a 6-week period 5 times to the designed protein, and then gave them a 6th dose, and then infected with the virus after that, and it still provided identical prophylactic protection. So these small proteins do not appear to be very immunogenic. Now, we expect, I didn't show it, but we know that those large icosahedral particles actually do elicit a strong immune response, and we're taking advantage of that for vaccines. So if you put proteins on the surface, you get very strong immune responses to them because they're presented in multivalent format. And so we actually have some very promising results with, for example, an RSV vaccine, because we get a very strong response against viral proteins when they're presented in that format.
Do you think there's a potential for a system with the control of something like DNA origami where you can pre-position desired functional groups at specific angles and then have the computer just fill everything in, or is it a little bit more?
You know, I think we're getting really close to that. And I showed that sort of DNA-like hydrogen bond sort of digital design of so we think we'll be what we're close to, we think, is almost an unlimited number of pairs of heterodimers, heterotrimers, and so forth. And so then at that point, building large structures is very DNA origami-like. But you have the real advantage that it's with proteins, so you can also incorporate functional groups as you like. So I think we're not quite there yet, but I think we're getting close to being able to just specify a geometry. Actually, we're working on that. We hope to have something ready in the new year where you can just specify a geometry, and then the thing will tell you, okay, you need to get these 12 genes and express the proteins, and it should form. So we're writing the code for that now. We haven't really tested it out yet.
Thanks for a good talk, David. The proteins, I've asked you this before, I'm sort of curious where things are now. The proteins you're designing are all in equilibrium conformations. How about where are you on non-equilibrium, energy utilizing?
Oh, yeah, that's right. Well, yeah, so where we, yeah. Motors, yeah.
Gizmos made out of proteins.
Right. So motors and machines is really high up on the list of things that we haven't done that we would like to do. So my own project has been to design proteins that basically have a set of sort of one-dimensional set of binding sites that are all identical to each other. And then we're making peptides and proteins that bind to single sites. So at least that way we should be able to get one-dimensional diffusion. And then we have to figure that to consumption of chemical fuel. And that part we haven't really figured out yet. But the idea is if you have things jumping between sites, maybe it's not so hard to go from there to fuel-driven movement. Right, thanks.